Hello, genetics class. Let's make some videos. Okay, let's talk about a monohybrid cross. So I think we left off about here. If not, someone can just email me and I will start earlier, but this is a great place to start. So a monohybrid cross, mono meaning one. So in this case, we're just doing big A times big A, little a, but it involves a true breeding. So true breeding is big A, not big A, little a. We would know that by selfing it, especially if it's a plant, we would cross it with itself over and over again, or we would cross it with some little a, just to see what our ratio is. But mostly you would cross it with another true breeding plant, or you would self it. Self meaning a plant where you just cross it with itself. This reveals how one trait is transmitted from generation to generation. Using this big A times little a, you're going to get 100% big A, little a, right? Heterozygous. Okay, so we have our original parents, dwarf, tall versus dwarf. We're going to breed those two, DD times little d, and this F1 generation is all going to be, I'm going to move me maybe over here. This F1 generation is all going to be big D, little d. You can do that Punnett square if you want, but basically you're taking, there are no other options. You get the big D's from one parent and the little d's from other parent, and everything is going to be big D, little d. Then for the F1 cross, you're going to self them. Basically, you're going to take this F1 cross and you're going to look at big D, big D, little d, and you're going to cross those. And you're going to get these gametes. It could give you this one or this one. It could give you this one or this one. So now we've made it down here. We're going to cross. So this is just a recap. We had tall versus dwarf, true breeding, remember? And from there we crossed those two and we got all big D, little d. Now we're going to self those, right? We're going to take big D, little d, big D, little d. In genetics, it helps very much if you try not to think of like the grossness of just like breeding the brothers and sisters. Think plants, don't think humans. It gets confusing. Just think plants. They breed with the one next to you, or you can make it breed with itself. So essentially, when we get to this generation now, this, we're bringing it down here. This plant could give the big D or the little d. This plant could give the big D or the little d. And we get all these possible options. Could be tall but heterozygous, could be dwarf. Okay, so that's the entire monohybrid cross that Mendel was doing. Here again, a reciprocal cross. Here it is using the Punnett square. So you have a D comes here, a D, big D here, little d here, little d here. Now we go this way. Big D here, little d here, little d here. Do you see how I did that? You can fill in the genotypes and phenotypes. And now you get 3/4 tall, 1/4 dwarf. This is should be a review. The results of a monohybrid cross are not sex dependent. We're not there yet. Basically, it's a 50-50 chance that you'll get either one of those, the big D or the little d. We're not including sex linkage yet. These are called particulate unit factors. Basically, the factors ended up being genes. Mendel didn't know that, of course, and genes serve as the unit of heredity from generation to generation. Unit factors, what are unit factors? They're basically genes. Dominance recessive. I'm saying this because I think it's a review. If it's not, I'll tell you now. So dominance, pair of two unlike unit factors for single characteristics in an individual. One unit factor is dominant, the other is recessive. So the dominant trait is the one that you see if both of them are, if you have a big D and a little d, the dominant will be the one that takes over. It will be the most predominant. So you're going to have Dark hair is dominant over light hair. Brown eyes is dominant over blue eyes. Meaning, if you have a gene that's a big E, little e, the big E is the dominant characteristic. If you don't know, you would have to self or cross a lot of plants to figure out which one of those is dominant. In Mendel's case, the darker color P was dominant to the lighter color P. And that's almost always the case because its pigmentation covers up the lack of pigmentation. And so when I say dominant versus recessive, I mean the characteristic that sort of masks the recessive, or the one that doesn't show up phenotypically. Segregation. Paired unit factors segregate, separate randomly during gamete formation. Who remembers gamete formation? It's meiosis. Yes. Mitosis creates exact pairs. Meiosis creates gametes, egg and sperm, and they only have one copy. And that is what makes genetics possible. Meiosis. Phenotype. So again, the physical expression of traits, the genetic makeup. Is it tall? Is it short? Is it red? Is it blue? Is it wrinkled? Is it smooth? Versus genes, or the unit of inheritance, found in alternative versions called alleles. The genotype is the allele makeup. Is it big D, big D? Big D, little d, little d, little d. You read the genotype and the phenotype is revealed. So there's a connection between genotype and phenotype. Always read the question, because I may ask you again for phenotype, and if you do genotype, it's going to be a little bit more work. Organisms inherit 2 alleles for each trait, 1 from each parent. Sex linkage in this case is interesting because if you are a girl, you got an X chromosome from your dad. If you were a boy, you got a Y chromosome from your dad. Your males have an X and a Y. If you're a girl, your dad gave you his X chromosome. If you're a boy, your dad gave you your Y chromosome. This is just how biology works. Homozygous genotypes have identical alleles. So basically, big D, big D, big D, little d. Homo, same. So they have the same alleles. So it can be homozygous dominant, which is this one, homozygous dominant, or homozygous recessive, which is this one. Heterozygous, different genotypes, 2 different alleles. Big D, little d. This just has to be vocabulary that you get down because these are the kinds of questions we'll have. Punnett square allows genotypes and phenotypes to result from across, and you visualize it. We're only going to do monohybrid Punnett squares, and then you'll just do the product rule from there. Vertical columns represent female parent, horizontal columns represent male parents. I do a lot of plant research, so sometimes I just put them out there, but I will try and be better about male versus female when we get to sex linkage. You will have to keep track of that. Again, here's our Punnett square. We've done monohybrid crosses. Test cross of one characteristic. Determine if the individual displaying a dominant phenotype is homozygous or heterozygous. So these are going to be backwards questions. I may ask you things about a parent. What do you think the parent is of this or that? So if you take, let's say you have a homozygous tall individual, and you are like, hey, I got this tall plant, and I don't know if it's homozygous or heterozygous. So what are you going to do? You're going to breed it with a dwarf. If you get all tall, then you know it's homozygous. You breed it over and over again, and you're like, every single one of these 1,000 plants is all tall. It has to be homozygous. But let's say you breed your tall plant with a dwarf plant, and you get some that are dwarf, you know it's heterozygous. Do you see how I did that? You have to be able to do this because it's a test cross, and it could be a question where I say you have a tall red plant and you breed it with a dwarf plant and you get half tall, half dwarf. What is the genotype of the parent? Those are the kind of questions that are going to come at you. I'll try and put some of those on a quiz so you can get used to looking at them.